Can everyone hear me all right? Great. So I'm going to be talking about my PhD research, which is looking at microbes in peatlands. But before I do that, I just wanted to kind of introduce myself quickly. So yeah, I'm from Newcastle, but I did my undergrad in biology and master's in ecology down in York before moving back home, where I worked as an A-level biology tutor for a few years. And during that time, I spent quite a lot of time volunteering for Northumbrian Wildlife Trust. And one of the things I really enjoyed and felt like I learned a lot from was getting involved in their peatland restoration work. And that's kind of what led me to this PhD. So before I talk about microbes, I thought I'd introduce peatlands and peatland restoration very quickly. So firstly, what is peat? Well, peat is formed from the buildup of partially decomposed plants, and that happens in very waterlogged conditions where there's not much oxygen and usually it's quite acidic. Peatlands are just areas of land with a naturally accumulated layer of peat at the surface. And globally, we have all sorts of different peatlands. So in the tropics, kind of around here, you get a lot of peat swamp forests. But further north, you get permafrost peatlands and tundra and these boreal forested peatlands. But in the UK, we basically just have bogs and fens. And essentially all the difference between those two is that bogs get their water from the rain, from precipitation, whereas fens get it from the groundwater. My research is mostly focusing on blanket bogs, which are a type of peatland that form kind of over large areas of both flat and sloping land, and they basically are like a blanket over the surface. And yeah, they're kind of generally quite miserable places if you describe them in too much detail. You're sort of like on the top of a hill, you might spend the whole day just in a cloud. In the summer, there's usually quite a lot of midges, but they're great, they're really interesting, and peatlands kind of more generally are very important. So one of the main things is to do with the climate. So peatlands cover just 3% of the Earth's land surface, but they store a third of all the soil carbon, which is more than like all of the forests in the world combined. And the way they can do that is that when plants grow, they photosynthesize, and they take carbon dioxide out of the atmosphere. And normally, when they die, they will decompose and like release that CO2 back into the atmosphere. But in peatlands, because it's so wet and it's so acidic and there's so little oxygen, they tend not to decompose. And so that carbon dioxide gets trapped and stored in the peat instead. And that's usually where the sort of focus of peatlands is. But it's also, they're so important for so many other reasons. They can help regulate water quality. So in the UK, about 70% of drinking water comes from these like upland, peatland-dominated catchments. They're also very great at sort of slowing like the flow of water down a catchment, and that means they can help with like flood risk management. They're also very wet, and that makes them unique environments, unique habitats, and so they're really great places for different biodiversity. You get a lot of different peatland specialist plants, like sphagnum mosses and cotton grasses, and carnivorous plants that, like sundews and things. There are a lot of insects, so you often see dragonflies and butterflies, and a lot of birds rely on them for breeding as well. And on top of those like 3 core areas that I've got up there, they're also great at preserving things, so they're interesting for archaeology and culture. Like I know a few artists that kind of all their work is to do with peatlands. But the problem is, like with a lot of things, human activities have degraded peatlands. In England, just 13% of peatlands are in like a near natural condition. And that is due to things like pollution and burning and peat extraction for fuel and for horticulture. They have also, especially in the uplands after World War II, been drained by cutting these ditches, these grips, that was to sort of improve, very unsuccessfully, productivity for agriculture and grazing. And all those different human impacts mean that peatlands dry out, they become less suitable for those specialist plants, they become more susceptible to erosion, And you get gullies like this forming where you have hags and areas of bare peat. This is actually quite a small gully. Some of them are sort of like 3 meters wide and like twice as tall as me. And when peatlands dry out, they also start releasing CO2 because there's more oxygen available and decomposition can happen faster. So they start contributing to climate change. Rather than acting as a carbon sink, they are instead a carbon source. And so because of all of this and our concerns about biodiversity loss and impacts on water quality, there's been a lot of work that's gone on into sort of restoring peatlands. And there are so many different methods that can be used, but kind of the most common thing is those grips that I was talking about, those drainage ditches, they get blocked. And that's usually using sort of a peat dam, and that means you get these pools of water that kind of build up behind them that help to like raise the water table level and sort of re-wet the surrounding area. And so that means it's more suitable for those peatland specialist plants. You also get things like sphagnum, like colonising these pools, and you get lots of different insects using them as well. As well as that, those gullies can be blocked. They can also be reprofiled. So those hags, instead of being at that like really steep angle, are made to be shallower. And that makes it easier for plants to grow. It means that you get less erosion and there's sort of more water held in the environment. And to sort of help plants colonise those like bare areas, we often help with the revegetation by either scattering seeds, or if there's like a site nearby that's got quite a lot of sphagnum or cotton grass, we translocate it. So we will take some of it, we'll break it up and sort of plant it in the bare peat. So there's lots of other things that can be done, but I hope that kind of gives a quick introduction to peatlands and peatland restoration. But where do microbes fit into all of this? Why should we care about them? Firstly, they're the ones carrying out most of the decomposition. So in a healthy peatland, that's going to be very slow and contribute to that formation of peat and storage of carbon. But in a degraded one, that it's drier, is happening much faster, and you're releasing a lot of carbon dioxide. Peatlands also release methane, and that is due to a certain group of microbes called methanogens, which just as a sort of part of their normal metabolism in anaerobic conditions, produce methane. There are also other groups of microbes called methanotrophs, which consume methane and can kind of like intercept the methane that's produced by the methanogens. And because methane is a really, really potent greenhouse gas, it's something like 28 times more potent than carbon dioxide, we really need to understand this balance between methane producing and methane consuming microbes. Especially because restoration is known to increase the amount of methane produced compared to like a degraded peatland. As well as these kind of roles in like greenhouse gases, peatlands can also help with nutrient cycling. Peatlands are quite a nutrient-poor environment, which is why you get carnivorous plants like sundews that use insects to get their nutrients. But microbes are able to recycle the few nutrients that are present and make them available for plants so they can actually grow. And we know that microbes have these important roles in peatlands, but there's a lot that we just don't know, we don't fully understand, and that's kind of where my PhD comes in. And so what I'm doing is working on a site in Northumberland called Whiteley Moor that's like right up on the border with Scotland, near Carterbar. It's kind of like if you are in Otterburn and just keep on driving, it's just there. And it's a really interesting site. So it is a national nature reserve, it's a site of special scientific interest, it's a special area of conservation, and it's owned and managed by Northumberland Wildlife Trust. It's a big site. It's 1,500 hectares, and half of it is that blanket bog I talked about to begin with. And over the past, decade or so, there's been a lot of restoration activity, especially in this area here. So doing all those sort of things like grip blocking and reprofiling gullies and things like that. But it's a big site, and there's still a lot of it that hasn't been restored. I mean, Whiteley, it's big, it's remote, restoration is expensive, and it takes time. There is actually restoration that's going on right now, like this season, and it's planned for the next few years. But for now, we've got the restored area and unrestored area. And what I'm doing is characterising and comparing the microbial communities that are present in those two areas. And the way I can do that is using environmental or eDNA. So I've been up to Whiteley a few times to collect peat samples, and then I will take them back to Northumbria and freeze them, and then when I get round to it, I will get them out in the lab and I will extract the DNA. So I will separate out the DNA from like everything else that is in the peat. And I can then get that DNA sequenced. And I will get lots and lots and lots and lots of different DNA sequences. And each of those sequences is like a barcode that is specific to a particular microbe. So once I've got all those DNA sequences, I can then compare them to a database of like known microbes. And that will allow me to identify which ones were present in the peat. I'm kind of in these like first two stages. I'm just at the start of my second year. So I've done some field work, I've done some lab work. I actually, just the last week, I got my like first sequencing data, but it's a tiny, tiny amount of sequencing data, but it's showing some promising, interesting things, but yeah, I've not got that today. But I do have one figure that was really interesting to me that was from a pilot study that was carried out actually like before I started my PhD. So, if I just explain this figure a little bit. So each of these points like represents the microbial community present in a peat sample. And these blue points over here are from the restored area, and these orange points are samples from the unrestored area. And essentially, the closer the 2 points are together, the more similar the microbial communities are. So for example, these 2 points, very, very similar microbial communities, but like this one and this one, those samples have very different microbial communities. Hopefully you can kind of see like why I was excited by this plot when I sort of first did it, because it shows that there is definitely some sort of difference between the microbial communities that are present in the restored area and the microbial communities that are present in the unrestored area, which means essentially there's a point of doing my PhD. I'm not wasting my time completely. But at the moment, I don't fully know what's kind of causing this difference. I have a couple of ideas. I'm sure it's to do with the fact that in the restored area, it's a lot wetter, whereas in the degrade area, it's very like dry and crumbly. So I have a feeling it's something to do with that. I have a feeling there's some sort of difference in the methane producing and methane consuming microbes, but I haven't quite spent enough time with the data just yet. But hopefully, as I do more field work and more DNA sequencing and get the data back and analyse it, I'll be able to start answering some of those questions. Because if we can understand Whiteley and peatlands on this very, very small, like microscopic scale, it will hopefully allow us to better understand the bigger picture impact that microbes might be having on water quality and the climate, and also just how restoration might be influencing these different processes. And hopefully that will allow us to use microbiology, use microbes and eDNA to monitor the changes in peatlands as restoration occurs, and also start thinking about, is restoration being successful? That's obviously for a couple of years down the line, but yeah, so far my PhD is there's some promising data, but yeah, I might be back in a couple of years or something to talk more about that, but for now I just wanted to say thank you for listening, and also thank you to my supervisors and to Northumberland Wildlife Trust and Northumberland Peat Partnership as well for their support with this work. So yeah, thank you. And any questions?